In this video, we'll be looking at cell cycle in somatic cells. In the cell cycle, there are three main stages, and those are interphase, mitosis, and cytokinesis. In the previous video, we looked at mitosis and cytokinesis, so in this video, we'll be focusing on the interphase. The interphase is the first stage of cell cycle. The interphase has three main stages, and those are growth phase 1, synthesis, and growth phase 2. In short, we can refer to these as G1, S, and G2. First, in G1 stage, all the cell organelles multiply by 2 in number. So that would basically mean if there are 3 mitochondria, then it would become 6 mitochondria at the end of this stage. This multiplication applies to all cell organelles except the cell membrane, nucleus, and the DNA. This stage is crucial because the end goal of cell division is to create 2 identical daughter cells. So we would want to multiply the cell organelles that are in the parent cell so that we can give half to each cell at the end. The next stage in the interphase is the synthesis phase. In the synthesis phase, the cell DNA is replicated. I'll be explaining synthesis phase in a little more detail in another video, but for now, the main function of this stage is to duplicate the DNA of the cell, so that one DNA can be given to one cell and the other one can be given to the other cell. Once the synthesis stage gets over, the cell will go on to growth phase 2, which is G2. In the G2 phase, the cell fixes all the errors and mistakes that occur during the first 2 phases. Generally, a lot of errors tend to occur during the G1 and S phase, so they're all fixed in the stage. A protein that's generally used to fix the DNA is known as P53. G2 is also referred to as a proofreading phase, since it checks if everything is perfect or not. This occurs in an ideal situation, but the cell is not in an ideal situation. The cell will not go through the cell cycle. Instead, the cell will go through a phase known as G0, which is growth phase 0. This occurs before the cell goes through the synthesis phase of the cell cycle. A cell can go through the G0 phase for 2 main reasons. One is an external factor and the other one is an internal factor. The internal factor is the genetic information of the cell. By that I mean if the cell DNA says that the cell should go into G0 and should not divide, then the cell won't do that. I'm sure at this point you all know that our body has different types of cells, like stomach cells, liver cells, skin cells, and many more. Some type of cells can actually have genetic code that instructs the cell to go towards the G0 phase. For example, this occurs in the neurons. You might have heard people say that our brain develops a lot when we are young, but after that, the brain doesn't develop anymore and only becomes weaker with increasing age. This is because neurons are not produced by our body after a certain age. And these neurons that are produced do not divide, which means they are in G0 phase. They are always present in G0 phase, which prevents them from dividing. And this decision is taken by the cell because the gene that is active in neurons say that the cell shouldn't divide. And this is why it's an internal factor, because the DNA is present inside the cell and that codes for the cell to go through the G0 phase. The other cause is external, which is basically caused due to conditions outside the cell. We all know that our cells need energy to do work, and that energy comes from the nutrients in our blood. Cell cycle is also a kind of work for our cell. In fact, during cell cycle, the cell does a lot of work since it has to replicate itself. If there aren't enough nutrients for the cell that could be used to produce ATP, then the cell can be diverted into the G0 phase. The cell will be in G0 phase until the cell gets enough nutrients to continue with the cell cycle. This is all for this video. Thank you so much for watching. If you like this video, then consider subscribing and liking this video. Bye.